Welcome back to the Deep Dive, your ultimate shortcut to being truly well-informed about the breakthroughs shaping our world. Today, we're plunging into a field that's not just transforming, but truly redefining our understanding of life itself. AI in biotechnology. Imagine, for a moment, your own genetic code, this vast, intricate instruction manual written in billions of chemical letters. And here's the kicker. Hidden within that immense blueprint, tiny, often overlooked variations can hold the secret to your health, your predispositions, or even the root cause of a disease. For decades, deciphering this code, making sense of that sheer volume of data, and understanding what these minute changes actually do. Well, it's been one of the grandest, most elusive challenges in modern biology.
It really has. It's like having the most complex, beautiful symphony, but, 98% of the score is in a language no one quite understands.
Exactly, and the few notes we do recognize are only a tiny part of the performance.
And that's precisely where advanced computational tools are becoming not just indispensable, but utterly transformative. So this deep dive is AI in Biotechnology Overview Part 2.
Part 2, right?
In Part 1, we explored the incredible advancements represented by the Arc Institute's state model.
Ah, yes, state. That was fascinating, simulating how cells respond to treatments at the population level.
Yeah, using those learned single-cell embeddings. That was really about understanding the micro-level impacts on a macro scale, giving us this unprecedented window into cellular behavior.
Ah.
Today, though, we're shifting our focus from simulating cell behavior to something even more fundamental, truly understanding the nuanced language of our own DNA, especially those vast, previously, inscrutable regions.
That's a critical distinction. So our mission today is to unpack a truly groundbreaking paper from Google DeepMind, boldly titled AlphaGenome, Advancing Regulatory Variant Effect Prediction with a Unified DNA Sequence Model.
It's a big one.
Yeah, this is a big one. We're going to break down AlphaGenome's incredible capabilities, how it utterly smashes through previous limitations in genetic prediction, and what its emergence means for our fundamental understanding of the regulatory code of life. As always, our goal is to give you a powerful shortcut to understanding this tool, highlighting its most surprising and impactful features so you can grasp its significance without getting lost in the weeds.
It's really about moving beyond merely reading the genetic code line by line to genuinely interpreting it.
Interpreting it.
Yeah, understanding the subtle nuances, the hidden switches, the vast networks that determine everything from how our cells develop and function to, our individual susceptibility to complex conditions like cancer or autoimmune disorders.
Right.
This ability to interpret is where the real power lies.
Okay, let's unpack this by starting with the central biological challenge AlphaGenome aims to tackle: interpreting the impact of genome sequence variation. When most of us hear about genetic mutations, our minds immediately jump to those changes in genes that directly code for proteins, like a typo in a recipe.
Yeah.
Makes the final dish turn out wrong.
The classic view.
But here's a surprising fact that often gets overlooked. Did you know that over 98% of all observed genetic variation in humans is actually non-coding?
Over 98%, it's staggering.
Yeah, it means it's not the parts of our DNA that directly tell our bodies how to build proteins. For a long time, these vast regions were considered, junk DNA, because we just didn't understand their function.
And this is precisely why these non-coding variants are particularly challenging to interpret and why they represent one of the last great frontiers in genomics. They don't directly alter a protein's amino acid sequence. Instead, they can dramatically modulate vital genome properties, often in subtle but profound ways.
How so?
Well, think of it like this. Your DNA isn't just a flat string of letters. It's intricately packaged inside the cell's nucleus, kind of like yarn wound onto spools, creating this complex 3D structure.
Okay.
This packaging is called chromatin. Non-coding variants can alter how this DNA is meticulously packed, whether it's tightly wound up and inaccessible, basically locking away certain genes, or loosely open and ready to be read.
So controlling access to the genes.
Exactly. These changes in chromatin accessibility can directly influence which genes get turned on or off.
And the complexity doesn't stop there, I imagine.
Not at all. Beyond the physical packing, these variants can also affect what we call epigenetic modifications.
Epigenetics, those are like the markers on the DNA.
You can imagine them as little sticky notes attached to the DNA or its associated proteins, yeah. They signal whether a gene should be turned on, turned off, or even turned up or down in volume.
So a tiny non-coding variant might change where one of these sticky notes could attach.
Precisely. Altering gene activity just like that.
Wow.
And then there's the 3D chromatin conformation. This is how the DNA actually folds up in the nucleus, bringing distant regions of a genome physically close together.
Like DNA origami.
Kind of. Think of it like a vast, incredibly intricate origami structure. A non-coding variant can alter this folding, disrupting crucial long-range interactions between a gene and its distant regulatory elements, like enhancers.
Enhancers, those are the switches often far away, right?
Exactly. So this folding change could potentially silence a gene or maybe turn it on in the wrong place or at the wrong time. This is where a lot of the magic and the mystery of gene regulation really lies.
It gets even more layered, I bet.
You bet. These non-coding variants can also influence messenger RNA or mRNA availability by altering expression levels.
Meaning how much of a gene product gets made.
Right. Or even more subtly, they can modify sequence composition through splicing changes.
Splicing. That's the cutting and pasting of the RNA message, right? Moving the non-coding bits.
Exactly. Splicing is that incredible biological process where non-coding sections, called introns, are precisely cut out from a freshly made RNA molecule, and the coding sections, or exons, are seamlessly joined together to create the mature mRNA blueprint for a protein.
Like editing a film, cut the junk, keep the good takes.
Perfect analogy. But a tiny change in a non-coding region, say in an intron near a splice site, can mess up this intricate cutting and pasting.
Leading to what? Faulty proteins?
Yeah, faulty or even completely missing proteins, or maybe a protein that has an entirely different function because an exon got skipped or an intron got left in. It can be catastrophic.
So the challenge then becomes immense, given that over 98% of observed variation is non-coding.
Right. Globally characterizing the complex cascading effects of this vast majority of variants remains completely intractable without powerful computational predictions.
We just can't do enough experiments.
Exactly. We can't cover every possible variant and its myriad effects across all cell types and tissues. This is precisely why AI is not just helpful here, it's absolutely critical.
It's the only way to move from correlation to causation.
Pretty much. It's the only way we can hope to really understand cause and effect in the vast landscape of the human genome.
Precisely. And that's where computational methods really shine. They learn these incredibly complex patterns from vast amounts of experimental data, allowing them to predict and then help us explain the effects of specific genetic variants. One crucial class of these methods are what we call sequence-to-function models.
Sequence-to-function.
Okay. Simply put, these models take a raw DNA sequence as input, just a long string of A's, T's, C's, and G's, and then predict what are known as genome tracks.
Right, genome tracks. To clarify that for you listening, think of it like a highly detailed, multi-layered map of the genome.
A map.
Yeah. Imagine each DNA base pair, each letter in the genetic code is associated with a specific value. This value might show you things like, say, how many DNA fragments were counted in a sequencing experiment at that specific point, or maybe the signal strength from an assay done in a particular type of cell or tissue.
So it's mapping experimental data onto the genome sequence.
Exactly. And these tracks are incredibly diverse, painting a comprehensive picture of genomic activity. They can cover everything from gene expression levels, using data like RNA-seq, CAGE-seq, or ProCap, to detailed splicing information, such as the precise locations of splice sites, how often those sites are used, or even splice junctions, the exact points where the cuts and pastes happen.
And it goes further, right?
Yeah.
What else can these tracks show?
Oh, yeah. They also map DNA accessibility using methods like DNA-seq or ATAC-seq, which tell you where the DNA is open enough to be interacted with by regulatory proteins.
Open for business, basically.
Kind of. Then there are histone modifications, measured by ChIP-seq. Those are those sticky notes we mentioned, indicating active or repressed regions.
Right, the epigenetic marks.
And transcription factor binding, also via TF-TYP-seq, showing exactly where crucial regulatory proteins attach to the DNA. And finally, and crucially, chromatin conformation, using data like Hi-C or Micro-C, which maps how DNA physically folds in 3D space, revealing those important long-range interactions.
Wow, that's a lot of different layers of information.
It is, and so what here is profound. Successfully trained sequence-to-function models can accurately predict these experimental measurements directly from an input DNA sequence. And the real utility, the core breakthrough that AlphaGenome truly elevates, insights comes when you compare the predictions made from a reference sequence, your standard human genome,
Okay.
to predictions from an alternative sequence containing a specific genetic variant.
Ah, so you compare the normal prediction to the variant prediction.
Exactly. By doing this direct comparison, these models can effectively predict the molecular effects of those variants, telling us with remarkable precision what functional changes they might cause at a deep biological level.
So it moves us from just seeing a difference to predicting its consequence.
Precisely. It moves us from mere observation to powerful, actionable prediction.
Okay, so if that's the ideal, let's unpack where previous deep learning models fell short.
Yeah.
Before AlphaGenome came along, they faced these two fundamental trade-offs, right, that really constrained their ability.
Big constraints.
It was a classic pick two scenario, you said. First up was the long-range versus resolution dilemma.
Indeed, it was a persistent challenge, a classic tug-of-war in computational genomics. You had models like Splice AI, BPNet, ProCapNet, Apparent.
Yeah.
These were truly excellent at providing base resolution predictions, meaning they could pinpoint changes down to a single DNA letter, which is crucial.
Precise.
Super precise. But they were fundamentally limited to short input sequences, typically 10 kilobases or less.
Only 10 kilobases. That seems tiny in the grand scheme of DNA.
It is. And it meant they often completely missed the influence of distant regulatory elements, like enhancers located tens or even hundreds of kilobases away from a gene.
So they couldn't see the forest for the trees almost.
Pretty much. Imagine trying to understand a complex circuit board by only looking at one tiny component at a time. You'd never grasp how the whole system works.
So on one hand, you had incredible detail, but a very narrow view.
Yeah.
What was the other side of that coin?
Conversely, models such as Informer and Borzoi represented the long-range side. They could process much longer sequences, ranging from 200 kilobases up to 500 kilobases.
Okay, that's much better context.
Yeah, allowing them to capture that broader genomic context and those important distant interactions. But they achieved this at a significant cost. They had to reduce their output resolution.
Ah, there's the trade-off.
Right, often down to 128 base pair or even 32 base pair bins. This binning essentially blurred fine-scale features that are absolutely critical.
Like individual splice sites.
Exactly. Individual splice sites, the exact locations where transcription factors bind, single base pair changes, and crucial motifs. All that got blurred.
So you had a wide-angle view, but one that lacked sharp focus.
That's a perfect way to put it. It was a significant limitation, forcing researchers to constantly choose between this zoomed-in, high-detail view that missed the big picture or a broad contextual view that lacked the precision needed for granular insights. You just couldn't really have both in a single model.
Exactly. You're always compromising. And then there was the second fundamental trade-off.
Yeah.
Diversity versus specialization.
This was another major hurdle. Many state-of-the-art models were, and honestly still are, highly specialized.
Specialized how?
Well, for instance, Splice AI is phenomenal specifically for splice site prediction. BPNet excels at modeling chromatin accessibility locally. Orca is designed to model 3D genome architecture, those contact maps we discussed.
So really good at one specific thing.
Incredibly powerful within their specific niche, yes, but they are fundamentally insufficient on their own to capture the truly diverse molecular consequences that a single genetic variant can have across multiple modalities.
Right, because a variant might affect accessibility and splicing and expression.
Exactly. Imagine a single genetic variant doing all that. With specialized models, you'd need multiple separate analyses, potentially running different software, maybe even needing different input data formats. It's a headache.
A real patchwork approach.
Yeah, and even within a single modality like splicing, specialized models might only predict splice sites, but not splice junctions, or that intricate competition between different potential splice sites.
Right. It felt like a piecemeal approach to a problem that's inherently interconnected. But then we started seeing models like DeepCA, Senji, Informer, say, Borzoi.
The generalists started emerging.
These generalist models began to demonstrate the utility and practicality of multimodal approaches, allowing one model to handle several tasks at once. They offered efficiency, and their learned general sequence representation could even be fine-tuned for new unseen tasks, which is incredibly useful.
Definitely a step forward. However, the drawback was that these more generalist models often lagged behind their specialized counterparts on certain tasks, especially those requiring very high precision like splicing.
Ah, so they were broader, but maybe not as deep in specific areas.
Kind of. Or they simply lacked particular modalities altogether, like those crucial 3D chromatin contact maps.
So scientists still needed to juggle multiple models.
Often, yes. They needed to juggle multiple different models and their associated data formats to piece together a comprehensive understanding of a variant's effects. It created significant friction in research workflows, really slowed down discovery.
Yeah, it sounds like the community was searching for that universal translator for the genome, but it was stuck in these unavoidable trade-offs.
Pretty much. Until now, maybe.
Here's where it gets really interesting. This is where AlphaGenome steps in, aiming to be the best of all worlds and utterly shatter those previous trade-offs, fundamentally changing how we approach genomic interpretation.
Precisely. AlphaGenome is presented as the singular, unified model that fundamentally integrates multimodal prediction, long sequence context, and base pair resolution into a single, cohesive framework.
All three: long context, high resolution, and multiple data types.
All three. And what's truly impressive is its input capacity. It takes a massive 1 megabase, that's 1 million base pairs, as its input.
1 million bases. That's enormous context.
It is. And from that single input, it then predicts an incredibly diverse range of genome tracks across numerous cell types. And critically, it often does so down to single base pair resolution.
Wow. So it eliminates that need to compromise on either length or precision.
Exactly. It's like finally having that microscope that can see both the entire forest and the individual leaves with perfect clarity all at once.
And it's not just about covering more ground, it's about depth, too, particularly in splicing, you mentioned.
Yes, particularly in splicing. AlphaGenome includes a novel splice junction prediction approach, working seamlessly alongside splice site usage prediction. This gives researchers a far more complete and nuanced picture of splicing events than was previously possible.
A fuller picture, not just predicting the sites, but the actual connections made.
Precisely. No more guessing. Now you have a unified, comprehensive view.
So how well does it actually perform? That's the key question.
And the proof is in the pudding, as they say. The results are truly remarkable. AlphaGenome achieved state-of-the-art performance on an astonishing 22 out of 24 genome track prediction tasks.
22 out of 24.
But even more critically for understanding disease, it outperformed existing models on 24 out of 26 variant effect prediction tasks.
Okay, so it's not just predicting the features, but predicting how variants change those features.
That's the crucial part, predicting how genetic variants actually change those features, which, as we discussed, is the ultimate goal for understanding their biological impact and linking them to human health and disease.
That is a game changer for diagnostics and research.
Absolutely.
And you mentioned accessibility, that Google DeepMind is making tools available.
Yes, and this is always an exciting part of these breakthroughs. They are not keeping this locked away in a lab. They are providing tools for making genome track and variant effect predictions directly from sequence.
So it's designed to be a practical tool.
Exactly. It's designed to be a practical, accessible tool for you, the researcher, the clinician, the curious individual, potentially transforming how we all approach genomic interpretation. This accessibility is almost as significant as the model itself.
That really lowers the barrier to entry.
Dramatically.
Yeah.
It lays the groundwork for widespread application across academia, biotech, and potentially clinical settings.
So how does it actually do all this? Unifying multimodal prediction, long context, and base pair resolution. That sounds like an incredible feat of engineering. Let's dive into AlphaGenome's inner workings, starting with its architecture.
Okay. At its heart, AlphaGenome employs a UNet-inspired backbone architecture.
UNet, like the letter U.
Exactly. If you're not familiar with UNets, think of them as neural networks that are exceptionally good at first compressing information into a dense, abstract representation, kind of distilling the essence of the input.
Okay, squeezing it down.
Right. And then skillfully expanding it back out to produce a highly detailed, high-resolution output. This makes it particularly effective for tasks where you need to map complex inputs, like a long DNA sequence, to equally complex, detailed outputs, like precise signals along that sequence.
Like reconstructing a detailed map from key features.
That's a great analogy.
So breaking it down simply, it processes that massive 1 megabase DNA input. First, there's an encoder pathway.
Correct. This pathway systematically takes that raw DNA sequence, starting at 1 base pair resolution, and progressively downsamples it, compresses it into more abstract embeddings at a coarser 128 base pair resolution.
Got it. Then what?
After that, a crucial transformer tower takes these compressed embeddings and processes them.
Transformers. Those are good at long-range dependencies, right?
Exactly. This is where those critical long-range interactions across the entire 1 megabase context are modeled. This is where the model understands how a genetic switch far away might influence a gene.
Okay, connecting the distant dots.
Precisely. Finally, a decoder pathway mirrors the encoder's job in reverse, upsampling those processed embeddings back to the original 1 base pair resolution, restoring all that fine-grained detail.
So it's this elegant dance between broad context and pinpoint accuracy.
It really is. And within that architecture, there's a clever combination of neural network components. It uses both convolutional layers.
Which are good for local patterns.
Exactly. Excellent at modeling local sequence patterns for fine-grained predictions. Think of them like small focus filters scanning the DNA for specific motifs or patterns. Highly effective for local regulatory signals.
On the other port.
Then it uses those transformer blocks, which are adept at capturing coarser but longer-range dependencies, like how an enhancer far away might physically interact with a gene's promoter.
So combining the strengths of both.
Right. And the beauty is that the model produces two types of outputs from these rich, learned representations. One-dimensional embeddings at both 1 base pair and 128 base pair resolutions used for linear genome predictions like expression or accessibility. And crucially, it also generates two-dimensional embeddings at a 2048 base pair resolution, specifically for spatial interactions, allowing it to generate contact maps, showing how DNA folds and interacts in 3D space.
Ah, so it predicts the 3D structure, too.
It predicts the contacts that inform that structure, yes.
Yeah.
Which is vital.
Now, processing 1 megabase of DNA at base pair resolution. That's not just computationally intensive. It sounds like a moonshot. How do they manage that?
Yeah, it's a huge computational hurdle. They managed it using an ingenious technique called sequence parallelism.
Sequence parallelism, like splitting up the work.
Essentially, yes. Imagine you have a single incredibly long rope and you need to process it all at once, but no single machine can handle its entire length.
Okay.
What you do is split that rope into segments and assign each segment to a different worker, all working simultaneously. That's basically what sequence parallelism does.
So they split the 1 MV sequence.
They effectively split the entire 1 megabase input sequence across 8 interconnected tensor processing unit, or TPU-v3 devices. Each device processes a segment in parallel.
But wouldn't that miss information at the breaks?
Good question. To ensure that convolutional operations don't miss context at the segment boundaries, each subsequence is extended with a 1024 base pair overlap from its neighbors.
Overlap ensures continuity.
Right. After processing, these overlapping regions are simply trimmed away. For the transformer tower, it's a bit more complex as it needs info from all cores, but they used optimizations to do it efficiently.
That's brilliant, a real technical feat.
It really is. It means AlphaGenome can now process DNA lengths previously impossible at this resolution, allowing us to capture interactions that span huge genomic distances.
Revealing hidden layers of control.
Exactly. Things that were previously invisible.
So from these rich, learned representations generated by the parallel processing and the UNet, how does the model actually generate its predictions for all those diverse genome tracks?
From these representations, output heads apply specific parameters for both human and mouse predictions across all the different modalities: expression, splicing, contact maps, etc.
Like specialized lenses on a universal knowledge base.
That's a good way to think about it. And what's particularly insightful is how it's trained. The total training loss is simply the sum of individual losses for each prediction type, without any special weighting.
No special weighting, so it learns everything equally.
It suggests its inherent generalist nature, yeah. It's learning all these diverse tasks simultaneously and finding common underlying patterns rather than being hyper-optimized for one specific task above others. This unified learning is a major part of its success.
So AlphaGenome wasn't just trained once and then unleashed. They used two sophisticated strategies to make it both powerful and efficient. It wasn't just raw power, but smart training.
That's right. Very smart training. The first strategy is pre-training, where the model learns directly from vast amounts of experimental genomic data collected from both human and mouse samples.
Learning from real biological data.
Exactly. It processes 1 megabase intervals, carefully centered around predefined genomic regions, and crucially, ensuring no data leakage between training and validation sets for unbiased results.
Okay, standard good practice. But you said there was more ingenuity.
Yes. The real cleverness here lies in the extensive data augmentation they employed during pre-training.
Data augmentation. Like showing it different versions of the data.
Precisely. Imagine teaching a model to recognize an object by showing it from every conceivable angle, under different lighting, slightly distorted. They did something similar for DNA sequences.
No.
They used shift augmentation, shifting input intervals slightly, making the model robust to small positional variations.
Okay.
They used reverse complementation, processing the DNA sequence and its reverse complement because genes can be on either strand, so the model learns symmetry.
Makes sense biologically.
Then they introduced random mutations, inserting a 4% rate of random nucleotide changes.
Simulating natural variation.
Yeah, but they went a step further, including structural variations, actual insertions, deletions, and inversions.
Wow, like real-world complex mutations.
Exactly. The number was sampled from a Poisson distribution. Lengths were uniform from 1 to 20 base pairs. This comprehensive augmentation makes the model significantly more robust to the kinds of variations seen in the real world, including rare or complex ones.
It's like teaching it to read a perfect book and decipher scribbled notes.
That's a great way to put it. It makes it much more resilient.
That's an incredibly thorough approach. What was the second strategy they used?
The second strategy is distillation. This is a fascinating technique.
Distillation, like distilling knowledge.
Exactly. Here, a single, smaller student model is trained to reproduce the predictions of an ensemble of 64 independently pre-trained, larger teacher models.
64 teachers, wow.
Yeah. Imagine training 64 expert musicians, each mastering a different aspect of a complex piece. Then, you train one prodigy student to perfectly replicate the combined performance of that entire orchestra.
How does that work, technically?
Each teacher model is loaded onto a different GPU, and the student learns by minimizing the difference between its own predictions and those generated by the specific teacher it's currently learning from.
And the benefit?
The benefit is profound. Efficient, high performance with significantly reduced inference cost. Instead of needing to run all 64 large teacher models for every prediction, which would be computationally prohibitive for widespread use, you only run the single compact student model.
Which has absorbed the knowledge of the whole ensemble.
Precisely. It's a masterclass in computational optimization.
So it's not just powerful in its predictions, it's also incredibly efficient and designed to be practical for real-world application.
Yeah.
That's huge.
It's a massive win for researchers. It means they can leverage this immense power without needing, supercomputing clusters right at their fingertips.
It truly opens the door for widespread adoption and accelerates discovery. Okay, let's zoom in on some of AlphaGenome's most impressive applications, starting with splicing. Just a quick reminder for everyone. Splicing is that fundamental, incredibly precise process where non-coding introns are removed, coding exons are joined, making mature RNA. Absolutely essential.
And disruptions can cause a vast array of diseases, from genetic disorders to cancers.
Right. So how does AlphaGenome tackle this?
This is where AlphaGenome truly provides an unprecedentedly comprehensive view. It's the first system of its kind to predict not just splice site probability and usage, but also novel splice junction prediction alongside direct RNA-seq coverage.
Novel junctions too, so it can spot entirely new splicing patterns.
Yes, it offers this truly holistic understanding of how a variant might impact the entire splicing landscape, where cuts are made, how often, whether new events occur. This integrated view is so powerful.
Can you give an example?
Absolutely. Consider a compelling real-world case, a 4-base pair deletion near the TAL1 oncogene.
TAL1 involved in leukemia, right?
Yeah. Yes. This variant is empirically known to cause exon skipping in tibial artery tissue, meaning an entire coding segment, a crucial sentence, is completely missed during splicing.
Okay, a known effect. Did AlphaGenome predict it?
It predicted it accurately across all levels of analysis. It showed a substantial reduction in the predicted usage of the affected exon splice site. The cut here signal was ignored. It predicted the complete loss of junctions that would normally link that skipped exon's edges. Critically, it also predicted the emergence of a new bypass junction that skips over the exon entirely.
Wow.
And finally, a strong decrease in predicted RNA-seq coverage for that exon, indicating the final mRNA product was indeed missing that section.
That's incredibly detailed. It predicted the whole chain of events.
It's a powerful demonstration of its accuracy for complex, cascading biological events with real clinical implications, like predicting exactly how a tiny typo in a blueprint causes a whole floor to be missing, and how the contractors then try to work around it.
That's an astonishing level of detail.
Yeah.
Any other splicing examples?
Similarly, AlphaGenome accurately captured a novel splice junction and an extended exon induced by another specific variant, GHR31.461262338JC.
And this was also seen experimentally.
Yes, this wasn't theoretical. This specific effect, a new cut-and-paste point and an elongated exon, had been observed in a real GTEx RNA-seq sample. AlphaGenome identified and predicted this complex, empirically observed event.
So it can find these subtle real-world changes.
Exactly, which is a significant advantage for identifying disease variants that act through novel mechanisms.
Now, how do they understand why AlphaGenome makes these predictions? You mentioned in silico mutagenesis,
ISM.
right, ISM. This method systematically predicts the effects of all possible single nucleotide variations within a given sequence region, like instantly trying every single letter change in a sentence to see how it alters meaning.
Testing every possibility computationally.
Yes. For example, an ISM analysis of exon 9 of the gene U2SURP and its flanking introns precisely highlighted recognizable splicing-related sequence motifs.
Confirming it understands the biology. It confirmed the model isn't just making statistical correlations. It has developed a sophisticated biological understanding of these fundamental regulatory elements and how changes affect splicing. It helps us understand the mechanistic basis of its predictions.
Okay, beyond splicing, non-coding variants can profoundly alter gene expression, influencing traits and diseases. How does AlphaGenome perform there?
This is another area of remarkable capability. It significantly improved the prediction of both the magnitude and the direction of EQTL effects.
EQTLs.
Yeah,
Expression quantitative trait loci, the genetic dimmer switches.
exactly. Compared to previous state-of-the-art models like Borzoi, AlphaGenome boosted the tissue-weighted mean Spearman row for coefficient prediction from 0.39 to 0.49.
A much stronger correlation with reality.
Much stronger. And even more critically for interpretation, for predicting the direction of the effect, does it turn expression up or down? It improved the mean sign ROC from 0.75 to 0.80.
That direction prediction seems crucial.
It's invaluable. It's not just saying a change will happen, but this change will make the gene more active or less active.
So what are the practical implications, say, for interpreting genome-wide association studies, GWAS?
Huge practical gains. GWOs identifies variants statistically associated with diseases, but often struggles to pinpoint the causal variant or its effect direction.
Right, the correlation is a causation problem.
Exactly. With AlphaGenome, at a 90% accuracy threshold for sign prediction, it recovers over twice as many GTEx EQTLs, 41%, compared to Borzoi's 19%.
Twice as many. That's huge for understanding GWO's hits.
It really is. It means AlphaGenome can pinpoint a specific variant's turning genes on or off with unprecedented accuracy, doubling our ability to understand the genetic roots of disease mechanisms from GWS data. And what's fascinating is that it often resolves the direction for largely non-overlapping sets of loci compared to conventional statistical methods like Kioser.
Meaning they're complementary, use both, get more answers.
Exactly. They collectively increase the total yield of loci with determined effect directions. Plus, it resolves about four-fold more credible sets in low minor allele frequency variants.
These rare variants that are hard to study.
Crucial, because they can be highly impactful but are often missed by population methods. AlphaGenome gives us a lens into these previously opaque regions.
So it's helping with common and rare variants. You also mentioned enhancer gene linking. How does it do there?
Right, linking enhancers those distant regulatory switches to the genes they control. AlphaGenome enables state-of-the-art enhancer gene linking.
Even without specific training for that task.
Exactly. When evaluated zero-shot, purely based on its general genomic understanding, it significantly outperformed Borzoi in identifying validated enhancer promoter links, especially for enhancers beyond 10 kilobases away.
That's impressive. It just understands these long-range connections.
It really seems to. It even achieved performance comparable to models explicitly trained on this task and cell line data. And when its predictions were incorporated into existing supervised models, it yielded a new state-of-the-art performance overall.
Wow. Okay, so putting it all together, AlphaGenome's true power seems to be its ability to analyze variants across multiple modalities simultaneously, getting the whole picture.
Exactly. That holistic view is where it truly shines, allowing for discoveries previously impossible. Take their virtual screening of that locus associated with T-cell acute lymphoblastic leukemia involving mutations upregulating the TAL1 oncogene.
Right, the cancer gene example. What did it predict there?
For a specific oncogenic mutation, CHR1.4723926.ccg, AlphaGenome provided this comprehensive, multimodal prediction that mirrored experimental observations beautifully. It predicted distinct increases in active histone marks like H3K27-axis, H3K4ME1, H3K4ME3 right at the variant site. consistent with experimentally observed neo-enhancer formation.
So the mutation created a new on switch.
Essentially, yes.
Okay.
Simultaneously, it predicted a decrease in repressive histone marks, H3K93 and H3K27E3, near the gene start site.
Removing the off switches.
Exactly. So the gas pedal is pressed and the brakes are released.
All from one change.
All from one change. It also predicted elevated active transcription marks, H3K36ME3, across the entire gene body, indicating a burst of activity and, critically, a direct predicted increase in tau-1 mRNA expression levels.
It painted the whole picture of upregulation.
A complete picture, undeniably showing its oncogenic potential at the molecular level, like watching a chain reaction unfold in slow motion.
And did the ISM analysis explain why?
It did. ISM revealed the variant actually introduced a MYB motif at the mutation position.
A binding site for a known activator protein.
Potentially, yes. The model predicted this new motif would increase tau-1 expression, chromatin accessibility, and H3K27 EC marks, just as seen experimentally. A direct mechanistic link?
Amazing.
It even highlighted a nearby ETS-like motif whose role is still unknown, but only seemed to affect tau-1 expression in the mutated sequence. This level of integrated mechanistic understanding is groundbreaking for pinpointing how mutations drive complex diseases.
It offers testable hypotheses.
Exactly, accelerating discovery.
This ability to see the whole picture, DNA packing, marks, binding, expression, splicing, all for a single variant. It's truly transformative for understanding disease mechanisms, moving beyond association to causal understanding.
And while AlphaGenome doesn't directly predict, say, whether someone will develop a disease,
The clinical outcome.
right, its predictions of gene regulatory implications are incredibly useful. By setting a high threshold on its quantitative scores, like predicted expression change, they could strongly enrich for causal variant candidates among controls.
Helping scientists zero in on the truly impactful variants.
Exactly, providing powerful functional hypotheses for variants whose mechanisms were unclear, complementing existing methods like conservation scores, which tell you a variant might be important, but not why or how. AlphaGenome adds the how and the why.
Okay, so the researchers didn't just build it. They meticulously pulled it apart to understand why it works so well. These ablation studies sound crucial.
They are. They offer vital insights for future AI and biotech. It's the science of understanding the science.
So what do they find? Starting with the impact of target resolution.
They've found that training with base pair resolution is incredibly important, especially for tasks needing fine-scale detail.
Like splicing and accessibility.
Exactly. Training with target tracks at 1 BP resolution consistently yielded the best results for those. Using coarser resolutions generally degraded performance for those fine-grained tasks. Detail matters during training.
But not always.
Interestingly, for tracks with inherently coarser assay resolutions, like contact map correlations or broad histone chip HESEC correlations, the model's performance was relatively insensitive to the target resolution used during training.
Which makes sense. You don't gain much by over-resolving coarse data.
Precisely. It informs how to efficiently train future models depending on the task.
What about sequence length? Does more context always mean better results?
Yes, generally, more context proved significantly better. Training with the full 1 megabase input sequences and evaluating with the full 1 MB context yielded the best overall results.
So training on long sequences is key.
It seems so. Models trained on longer sequences significantly outperformed those trained on shorter ones, even when the shorter trained models were given longer context for evaluation.
So the learning happens better with more context during training itself.
That's what it strongly suggests. It forces the model to learn those intricate, long-range regulatory relationships, like reading the whole book to understand the paragraph.
Right. But could it still work with shorter context for faster predictions?
Interestingly, yes. They found a model trained on the full 1 megabase could still perform remarkably well even when assessed with shorter inference contexts. This offers flexibility, maybe use a smaller window for speed if the task allows.
That's useful. Now, distillation, that teacher-student approach, how effective was it, really?
Yeah.
Was it a win for efficiency?
Distillation, especially using many teacher models they used, 64, proved highly effective and, was a major win.
How did the student compare to just averaging the teachers?
The single student models performed competitively with, or sometimes even surpassed, the average performance of several independently pre-trained models run as an ensemble.
Wow, really? And much cheaper to run.
Significantly reduced inference cost. Running one student model is far more efficient, orders of magnitude faster and less resource intensive than running and averaging 64 large models for every prediction.
That's a huge breakthrough for practical application. Makes it accessible.
Absolutely. It means the best knowledge can be disseminated widely without needing massive compute resources.
And finally, the core idea, multimodal learning. Is combining all these data types truly better than specializing?
The ablation studies strongly confirmed its value. The fully multimodal AlphaGenome model consistently and generally outperformed models trained on only single modality groups.
So integrating diverse data types really helps.
Yes, it confirms the benefit for learning shared, holistic representations. While some tasks, like accessibility prediction, could be done well with just accessibility data.
Others needed the full picture.
Right. Crucially, predicting EQTLs strongly benefited from the full multimodal input. This demonstrates the value of the unified approach for broad, high performance, especially for tasks integrating diverse signals.
It suggests the model gains a deeper understanding by seeing everything together.
Exactly. It highlights a more robust grasp of fundamental regulatory principles encoded in DNA. The sum really is greater than its parts here.
So, what have we truly learned from this deep dive into Google DeepMind's AlphaGenome? It's clear this is a truly significant step forward in deciphering the genome's regulatory code.
A huge step. An area historically incredibly challenging due to that complexity and the sheer volume of non-coding data. AlphaGenome offers this unified deep learning model that simultaneously predicts diverse functional genomic signals from megabase scale DNA, matching or even surpassing specialized models. Its ability to efficiently predict variant effects across all predicted modalities in a single pass is absolutely crucial for understanding variants with complex, multi-layered mechanisms like we saw with Tau1.
Right, seeing the whole cascade.
And that novel splice junction prediction feature provides a far more holistic and accurate view of splice-disrupting variants, vital for diagnosing many genetic disorders.
And the exciting news, the accessibility part.
Yes, it's not just a paper. Google DeepMind will make it available for non-commercial use via an online API and a Python SDK, with source code and weights available upon final publication.
That's huge for democratizing access.
It's a critical step for accelerating research globally. They also provide a comprehensive genome interpretation suite to facilitate exploration, streamlined variant scoring, ISM experiment.
They're making it user-friendly.
Exactly. It means researchers and clinicians worldwide will soon have incredibly powerful, user-friendly tools at their fingertips, enabling more precise, comprehensive, and efficient genomic interpretations than ever before. This is the future of understanding our genetic blueprint.
So thinking about AlphaGenome's unprecedented power to interpret those, once mysterious non-coding variants and its integration with other approaches, it really opens up new vistas. What new frontiers in personalized medicine or even disease prevention do you think could be unlocked by such a comprehensive understanding? How might this shift our understanding of what it means to be well-informed about our own unique genetic blueprint?
Well, it moves beyond just knowing you have a gene variant.
And knowing what it does.
Right, to understanding its precise regulatory impact. This raises an even more profound question. If we can predict the functional impact of nearly any genetic variant with such precision, moving beyond statistics to mechanism, how might this shape our entire approach to genetic screening for predispositions? Could it accelerate precision drug therapies by targeting specific regulatory mechanism?
Or even redefine health itself?
Maybe. Redefine our very concept of health in the coming years as we uncover the subtle genetic variations that influence well-being long before disease manifests. The implications? Well, they're truly immense and perhaps just beginning to unfold.